Department of Emergency Medicine, Institute of Inventon Medical in University of Pennsylvania, who will tell us about pressure and the yin-yang of the good and the bad of oxidative stress, and he'll show you, as he's shown in many of his wonderful publication paper, what's the meaning of, the real meaning of oxygen dose. Please.
Real meaning? Good afternoon, everybody. First, let me say thank you very much for inviting me to be at this wonderful meeting. And Rami, congratulations. It's been a great pleasure to know you for all these years and hope to see you at lots of future meetings. So the title Rami gave me, it was Pressure in the Yin-Yang of Oxidative Stress. And I only added the word oxygen because really that's what we're talking about predominantly with regard to the pluses and the minuses of a particular approach or therapy. So we'll get right into it, sort of a central issue, and this is something that people were dancing around really for most of the morning session as well. So oxygen, whether it's administered related to diving medicine or in the diving mixture or as a therapeutic tool, hyperbaric oxygen, we're basically talking about oxidative metabolism, a transient change in oxygen level in whatever tissue you want to talk about, and a response to that oxygen level. And the issue becomes, if you want to look at a long-term either benefit or detriment, to which side of the coin you want to be on, you're still asking the question, how does a transient change in metabolism cause anything that's long-term or meaningful, plus or minus? And I would maintain that it all revolves around production of reactive species, reactive oxygen or reactive nitrogen species. And now, of course, we talk about oxygen toxicity, and we'll talk about some of the details of that in the coming couple of slides. But nonetheless, yeah, we have to recognize that there's a downside to everything that we do as any therapeutic approach. There's oxygen toxicity. We try to avoid the toxicity while getting the benefit. There's benefits of hyperoxia in terms of modifying, if you will, vascular changes related to inflammation or more generally ischemia reperfusion, and that also includes bubble disorders, decompression sickness most specifically. These changes often happen in the minutes to hours of oxygen exposure. More long-term effects that take many days really to see a response is wound healing, which is a much larger part of these days at least, the sort of hyperbaric medicine world. Now, in terms of the subject, the yin-yang issue, basically these principles persist no matter which side of the coin you're on, and it all depends on dose, duration, and frequency. And that's where the crux of the matter would be in terms of one's, if you want to hone in on a particular response, you need to not only talk about dosing intervals, but you have to talk about the tissue that you're speaking of as well, because the biochemistry changes. So generally speaking, we're talking about reactive oxygen species, superoxide being sort of the representative of the group, and nitric oxide. And anytime one is exposed to hyperoxia, or even ambient pressure oxygen, you get production of superoxide radicals and an array of other secondary radicals, predominantly from mitochondria, endoplasmic reticulum, peroxisome, phospholipid metabolism generates free radicals, and a whole array of oxidases. The more oxygen, the more free radicals. Now, there's also new, relatively speaking, on the scene nitric oxide, produced by the metabolism of arginine and our friend oxygen to citrulline and nitric oxide by an array of 3 different forms of nitric oxide synthase. Now, an important sort of caveat here is this is not a parallel highway here where 2 things are happening independent of each other. Superoxide has a huge impact on metabolic rates of nitric oxide synthase. Similarly, nitric oxide, most specifically from mitochondria, has a major impact on mitochondrial oxidative phosphorylation, production of free radicals, production of ATP, predominantly as a negative modulator of mitochondrial function. So you get more of one, you get one response, you get more of another, you get another response. If you're talking about tissues that have, say, for example, nitric oxide synthase brand 2 or inflammatory nitric oxide synthase, all by itself, INOS2 generates concurrently nitric oxide and superoxide. And you can have a big difference of balance of the biochemical responses in different tissues. And that'll be a very important point in about 4 or 5 slides from now, and I'll try to remember to bring it up again. All right, so toxicity. Well, we all are relatively familiar with toxicity. It's something we live with all the time in terms of hyperbaric medicine. In terms of diving-related issues, one uses oxygen or elevated oxygen levels to decrease the duration and improve the efficiency, if you will, of decompression procedures. So it's true in this entire field that we have to live with the toxicity issues. In terms of pulmonary dosing is everything again. The longer the interval of time at any particular partial pressure, the higher the propensity for getting adverse effects in vital capacity, expiratory flow rates, or diffusing capacity measured with CO, particularly in sat diving. This is something of an issue. CNS, we're talking about seizures, we brought up a couple of times this morning. Typically thought to not be a real risk until you reach 1.5 ATA or so. Clinical protocols, we try to avoid seizures. And the overall incidence is about 1 to 3 in 10,000 cases. Then there's visual effects that are progressive in terms of narrowing of peripheral vision, myopia, which happens with repetitive treatments, and with really protracted huge numbers of treatments, I would submit inordinately beyond anything that's therapeutically relevant. One can get cataract formation, again, from oxidation of the proteins in the lenses of the eye. So that's the downside. Now, let's spend a little more time talking about the plus side for a couple of reasons. Again, HBO, oxidative metabolism, reactive species, short-term effects in terms of, if you will, in quotes, ischemia, reperfusion, inflammation, inflammatory responses, and wound healing. These are more of immediate effects. They happen during the oxygen exposure and persist for an interval of time afterwards. Some of the later things, you get an initial response to the hyperoxia that persists for many, many days. To decide a therapeutic intervention for a particular disorder, I would submit we have to have much better sense of mechanisms of action to be able to define dose, duration, partial pressure to use, when to intervene in the course of a particular disease. In the context of ischemia reperfusion, many of you know this sort of common story in terms of endothelial changes that happen during the interval where there's either a reduction or a complete cessation of blood flow. Blood flow is restored, but the endothelium has now been modified. It's not the same pipe that it was before the ischemic interval. And now, because of that change of the endothelium, neutrophils will now look at the endothelium as a foreign substance. They will begin to interact through, if you will, constitutive adhesion molecules, become firmly attached through adhesion molecules called beta-2 integrins. They become activated, they cause vascular damage, more endothelial injury, and a positive feedback loop that is invariably bad for you, leading ultimately to tissue injury or tissue death. Hyperbaric oxygen has a beneficial effect here. It's a discrete effect, though, based on dosing, particularly dosing. And hyperbaric oxygen can inhibit beta-2 integrin adhesion function and have a very beneficial effect in terms of ischemia reperfusion. The key here is when you're going to administer it. Our view was, well, okay, that's fine, but we first have to define the mechanisms of action. And here we go with this yin-yang of oxidative metabolism, if you will. Hyperbaric oxygen inhibits, we showed in years gone by, this happens in any array of rodent models. It also happens in humans. So this is data that's actually quite dated now. In terms of volunteers, we draw blood, we assess their beta-2 integrin neutrophil adhesion function, expose them for 45 minutes to 2.8 or 3 atmospheres, and immediately after that treatment, draw blood again, and adhesion function is virtually nil. However, 24 hours later, it's back to normal, which is key. You get a transient effect, which can be beneficial in terms of the primary pathological event. But if you had permanent beta-2 integrin inhibition, we call that immunocompromise, and that's not good for you either. So having a transient effect is quite critical in terms of positive without the negative. This is just a picture of what we're talking about. So this is a mouse neutrophil, but we've done the same thing with human neutrophils. We're looking at filamentous actin, the cytoskeleton of a normal neutrophil stuck to a glass slide. In order for beta-2 integrins to work, they are displayed across the surface of the membrane of the cell. They have to come together in a jargon term called capping in order to work in a coordinated fashion to stick. And that's a picture of capping of beta-2 integrins, and then a confocal overlapping picture. With hyperbaric oxygen, you get this gel of filamentous actin, profoundly different structure with a complete absence of capping and no adherence. So that's a picture of what's going on. The biochemistry gets a little bit more complicated. We'll sort of cover the highlights without putting you all to sleep with the details. This is our first paper on the function. But this now brings up once again the yin-yang issue. This is a discrete effect of neutrophils. Beta-2 integrins are found on essentially all circulating white blood cells. But beta-2 integrins on monocytes are not inhibited with hyperbaric oxygen. How come that should be? How could something so simple like hyperbaric oxygen have such a discrete effect? And it has to do with the physiology as well as the biochemistry. Packaged in the same primary granules of neutrophils is nitric oxide synthase, brand 2, and myeloperoxidase. And it's the concurrent production of reactive species, nitric oxide and hydrogen peroxide from myeloperoxidase. These agents interact to make more reactive nitrogen species that nitrosylate cytoskeletal actin. Nitrosylate simply means adding an NO group to the thiol, the SH of cysteines. And we've actually described this in this paper. There's 6 carboxy terminal cysteines that are modified by hyperbaric oxygen. And this actin S nitrosylation is what ultimately leads to the impairment of cytoskeleton and the transient inhibition of beta-2 integrins. This, however, is even more complicated. Hyperbaric oxygen is a very dynamic effect. You can expose cells for intervals of time and get a slightly different story. So here we're looking at nitric oxide production by neutrophils during hyperoxia. And what we see is this so this is the time the cells are exposed to elevated oxygen pressure. Transient marked elevation in nitric oxide production by the neutrophils, which only lasts as a peak for about 10 minutes and then drops to about half normal, or half of the elevated one sees it at the outset. So there's a biphasic effect here. What the heck is going on with all that? Well, it turns out that, again, there's a bimodal response mediated in large part by this cytoskeletal beta actin. It gets S-nitrosylated. VASP is a chaperone protein involved with cytoskeletal control. And it just turns out, we've published another paper, where VASP has a markedly higher affinity for S-nitrosylated actin. This binds, causes a very rapid turnover of short filamentous actin, basically, if you will, a cycling of the cytoskeleton, increasing short actin filaments, which then attract yet another scaffolding protein called focal adhesion kinase. That links here, and INOS brand 2 follows in suit. And it's with this linkage to actin that causes elevation of nitric oxide synthase activity, which turns out to be key to the HBO effect to drive beta-2 integrin inhibition. And here's a picture. So we're looking here at focal adhesion kinase overlapping with INOS, nitric oxide synthase brand 2. In this panels down here, we're looking at filamentous actin stained with phalloidin and INOS, looking again at the overlap. So you're looking here at the merged picture. So that's what a normal cell looks like. At 15 minutes, we see this peak INOS activity. There's a markedly higher fluorescence overlap, if you will, which is showing that all of these things are linking together. And this exactly overlaps with elevated INOS function. But it's only transient. It falls apart. And so by 45 minutes, there's no longer any marked elevation of INOS, and the cytoskeletal response is only transient. All this is sort of a sidelight, but it's bringing the point that all of these things are dynamic events that are happening. We can define each step individually, and they are all mediated by free radicals. You can look at other tissues. So here's a study we did some time ago looking at brain production of NO as a function of hyperbaric oxygen. And this is just looking at wild-type mice, mice with a knockout of eNOS or NOS brand 3, and this is NOS brand 1. And the point here is virtually all of the elevation of nitric oxide synthase production in the brain is mediated by type 1 neuronal nitric oxide synthase. You take that out, you take out, for the most part, all of the elevation of NO. Why does transient hyperbaric oxygen cause a persistent elevation of nitric oxide synthase activity? Biochemically speaking, that shouldn't happen. If it's merely a substrate phenomenon, it should happen only during the time of HBO and then go away. But it's not simply a dose response or a fueling of an enzyme with substrate. There's a secondary effect, and this is key, again, with the broad picture, if you will, the yin-yang of oxygen. A response to oxidative stress is antioxidant effects, generally speaking. You say, duh, we kind of know that's about to happen. But it's the real evidence gets into the fine points. One of the responses are heat shock proteins. And actually it's been known for quite some time. Heat shock protein 90, if it links up with nitric oxide synthase, markedly elevates its activity. And this is just showing, this is immunoprecipitation of brain. Looking at it, if you immunoprecipitate with HSP, you bring down much more nitric oxide synthase, and you can play this game both ways. Basically, the point is you can biochemically demonstrate a linkage between these enzymes. We see, and you can plot it any way you want, and there's an exact correlation between this linkage and nNOS activity elevation by HBO. So it's a secondary effect of the hyperoxia that drives the NO production in the brain. Similar sort of biochemistry happens in the vasculature. This is another study we did looking at aorta, looking at elevations of nitric oxide production in the aorta, which you might think is due to eNOS perhaps, but it turns out in this case, some of the major muscular vessels, it's an nNOS mediated phenomenon once again. Other people have done work along this line. Another group has looked at hyperbaric oxygen as a potential therapeutic intervention for acute myocardial infarction. And so here's the control animal, huge infarct zone, 1 ATA, really statistically no reduction of infarct zone, but 2 atmospheres, markedly better viability of muscle. And so this was the model they used. And basically they demonstrated that the functional mechanism for this benefit was HSP90 linkage, not here with NOS1, but with NOS3. Once again, it's sort of an overlapping phenomenon, but it's an oxidative stress response that actually drives persistent, if you will, therapeutic response to HBO. Now, changing subject. Separate from these acute effects, vascular effects, ischemia, reperfusion, let's talk about wound healing in a few minutes. So dogmatically speaking, we've known for decades wounds heal through so-called angiogenesis, meaning you cut yourself and the local resident endothelial cells right next to the wound proliferate, migrate into the void, make new blood vessels, rehook up, and heal the wound. And for the long time, that was thought to be essentially the only way wounds healed in adults. In neonates, there may be another way, but adults, this was the way things happen. We now, though, know that that's not true, and in fact, a large component, even in adults, of wound healing has to do with so-called vasculogenesis, which is a de novo process occurring when stem or progenitor cells, predominantly from the bone marrow, but also from peripheral sites as well, mobilize, meaning go into the bloodstream, migrate to the site of the injury, differentiate into an entirely new vascular channel, and involved also, there's a synergism here, in terms of healing the wound. This may happen in other tissue beds too, but primarily our data has to do with peripheral wounding.